it's my pleasure to introduce Elly Lynch. So I've known Elly for a number of years now. She's a well-experienced genetic counsellor and provides information and support to many patients and families who she sees, explaining the risk of genetic conditions. She currently works at the Victorian Clinical Genetics Service based at the Royal Children's Hospital, but she's worked in paediatric genetic clinics, adult neurogenetic clinics, as well as assisting coordination and conduct of outreach in Northern Territory Genetic Services, which I imagine is quite different to what you experienced in Melbourne going out back. Prior to working at VCGS, Elly Elly co-established the Clinical Genetics Services at Austin Hospital in Melbourne and worked as a unit manager from 2007 to 2016. So she's worked at a range of different hospitals, Royal Melbourne, overseas at the Royal Marsden Hospital in the UK, and also has worked for the Melbourne Genomics Health Alliance, which was a collaboration, or still is a collaboration, between 10 Melbourne hospitals and research institutes, working together to bring genomics into mainstream healthcare. And that alliance is still going, but Elly is no longer a member, and we miss her because I'm on that community advisory group. So welcome, Elly. Elly's going to do her presentation, and then there'll be a Q&A session at the end. Elly, would you be happy if people put up their hands or put it in the chat if they want to ask a question as you go through?
Yeah, of course. So I'm hoping to keep this as interactive as possible. I do have a formal presentation, but if people want to butt in and ask questions, just do so or put it in the chat. And Heather, let me know if something goes in the chat and I haven't noticed. But yeah, as Heather said, very happy to keep this as interactive and helpful for people as I can. So I'll share my screen. As I said, I do have some slides. Is that on presenter view, Heather? Yeah, great. Yeah, so thanks very much, Heather, for inviting me to speak. As Heather mentioned, I'm a genetic counsellor in Melbourne at the Victorian Clinical Genetics Service. I've been a genetic counsellor for over 20 years now and worked at a few hospitals across Melbourne, as well as with the Melbourne Genomics team. And I've had quite a bit to do with SWAN, particularly over the last few years, definitely referring families to SWAN and, you know, getting Heather's help sometimes with some of the families that we see who really do require some further support. Whenever I go to a party or I go to a new hairdresser or something and they ask me what I do and I say that I'm a genetic counsellor, I find that genetic counselling is not something that's particularly well known by the general public, and also not a profession that's often very well known by other health professionals as well. So even though it's something that I've been doing for a long time and I think the profession is growing, it's still a relatively small profession compared to other allied health roles in Australia and worldwide. And exactly like what Joel said, genetic counselling is really a communication process. So it involves, depending on the purpose of the appointment, it might involve interpreting family and medical histories to assess the chance of a condition occurring or reoccurring. So sometimes we see people, for example, who have a family history of cancer or who they themselves might have had cancer, and we're looking at the family history and assessing the likelihood of there being some sort of genetic or inherited cause for that cancer. Education is a big part of what we do. So we might educate people, hopefully in a simple way, in lay terms, about, for example, the natural history of a condition, the inheritance pattern. So whether a condition's dominant or recessive or X-linked. We use lots of pictures when we're explaining those sorts of things. We explain genetic testing, and often research as well, which I'll talk a bit more about as well. And I guess that we, as part of our role exactly is providing counselling to families. And that might be around decision making. So families might be making decisions about whether or not they want to have a test for themselves, or they want to have a test for their child. And I guess that we encourage people to make decisions that best fit with their own family goals, their cultural beliefs, their ethical preferences, and religious views. And hopefully, a lot of what we do also is to provide support to families, and that might be around adjustment to a genetic condition. Working at the Children's Hospital, obviously we see lots of parents of children who have been diagnosed, sometimes very, very young. They might be newborns who've been diagnosed with a genetic condition, which can come as a massive shock to people, and it's providing support, information, reassurance where we can, and I guess over time, as well as people's information needs change, just being there as someone who can provide support to that individual or that family. So some of I guess that there are some common questions that we get asked in appointments, obvious ones like, what is this condition? I've never heard of it. Can it be passed on? So parents might be having another child and they're wondering about the chances of another child having the same condition. If it is something that could be passed on, what their options are to avoid passing the condition on. For example, if they have other children, how, will they also potentially develop this condition? How could it affect them? Genetic testing, as we often talk about, is very different to other tests that people have, in that it can sometimes have a bit of a snowball effect. So rather than just a result affecting one person in a family, it might be a result that affects lots of members of the family, and family communication can be quite difficult. And so sometimes conversations involve how to approach other relatives about the chance of this condition potentially affecting them or their children as well. Do I need any tests, and when? Usually the first thing that we say when we are talking about testing a child is whether or not they need to have a blood test, because that's usually the one thing that the children are most anxious about when they come and see us, that they might need to have a blood test. And these days often we can do testing on saliva rather than blood testing for genetic testing. Obviously questions around how they or their family access services. We often ask to write letters of support for NDIS funding and so forth. Are there others with the same condition? So particularly if a family has a child who's diagnosed with a very rare condition, I don't need to tell this audience the value of peer support, but talking to another family who have a child or another family member with the same condition can be extremely helpful for some families. And sometimes the question around, is this my fault? Did I do something wrong? Was there something that I did during the pregnancy or that I didn't do during the pregnancy or before the pregnancy that caused this? That's a question that comes up often. And, I guess it's something that sometimes people feel anxious about asking us, but something that we can almost always reassure people that it's a genetic condition or a genetic change that's occurred is just something that generally happens by chance. So in Australia, there's 2 courses to study genetic counselling. So generally people have an undergraduate degree. They must have at least second year genetics as part of their undergraduate degree. And there are 2 Masters of Genetic Counselling courses. So one in Sydney at the UTS and one in Melbourne at the University of Melbourne. There's about 20 students in each Master's course. It's a 2-year course. It's quite competitive to get into the Masters of Genetic Counselling. There's an entrance, usually a genetics exam or quiz. There's an interview, and generally people who apply for the course are encouraged to have had some life experience as well. So not just coming straight from university and straight into a master's, but having done some volunteer work or having some relevant life experience. When people graduate from the course, they are associate genetic counsellors. And then people undertake a minimum of 2 to 3 years on the job training and apply for certification as a genetic counsellor. So that means that they are writing up case reports, they're doing simulated consultations, reflecting on practice and so forth. So there's a fairly arduous certification process that people have to go through the Human Genetics Society of Australasia to become a certified genetic counsellor. And a few years ago, genetic counselling became a self-regulated profession via what's called the Human Genetics Society of Australasia under the auspices of NASRAP. So there was quite a process that we went through in order to achieve that. So this slide is probably a little bit old, but in Australia, this gives you a bit of an idea of the spread of genetic counsellors across the country. It's a fairly small but growing profession. It's fair to say that it's generally female dominated rather than male dominated. I guess people work in different roles, and those roles are expanding. So people like me work hospitals across Australia, but there are more and more private genetic counsellors around now. So particularly in ultrasound practices, IVF clinics, some people who are self-employed are working privately. Laboratories across Australia are now employing genetic counsellors as well. So at the Children's Hospital, we have a laboratory genetic counsellor at the hospital, as well as with the Victorian Clinical Genetics Service. And their roles mainly involve helping other non-genetics health professionals with ordering and interpretation of genetic testing results. And I guess while genetic counselling might be part of the role of many different health professionals, trained genetic counsellors are the ones who have the specialist skills in genetic counselling. So across Victoria, these are the sort of main clinical genetic services that exist. So there's adult genetic services, paediatric services, familial cancer clinics, which are often quite busy, prenatal services, and then we also have regional clinics as well. So the VCGS, where I work, they travel to all of these places here and conduct regional clinics. And the adult familial cancer centres also travel to each of those places as well to conduct adult familial cancer clinics in country areas. So it can be a bit confusing, but VCGS is the genetics department of the Royal Children's Hospital. So even though we're part of the Murdoch Children's Research Institute, we all have honorary appointments at the Royal Children's Hospital, and we work with lots of different specialists across the hospital to conduct various genetics clinics. So one of the clinics that I'm actually involved with is the eye genetics clinic at the hospital, where we work with the ophthalmologists at the Children's Hospital, one of the clinical geneticists and myself, to see children who have genetic eye conditions. And there's lots of other specialist clinics now as well. So dermatology genetics, Tiong Tan runs a craniofacial clinic, there's a brain malformations clinic, heart, neuromuscular, and so forth. So there's, it seems that genetics and genomics is sort of infiltrating every single department of the hospital now as it's becoming more and more relevant. And that's just a bit of an old photo, but some of the genetic counsellors that work at VCGS at the Children's. Ivan Machoka, who's been around for a long time, is our senior genetic counsellor, and he leads the team of genetic counsellors there. We generally work as part of a team. So we have quite a big team at the Children's. We have our administrative staff who are the glue, who hold us all together and book all our appointments and keep us organised. There's a big team of associate and certified genetic counsellors. We have a team of screening genetic counsellors. So they're the team that are involved with all of the newborn screening that's done, all of the pregnancy screening, so the non-invasive prenatal testing or the combined first trimester screening results that the lab does. They have a whole team of counsellors involved in that. We have a team of clinical geneticists and genetic fellows. That's Martin Delaticki there, who's just stepped down as director of the VCGS service. We have a team of clinical assistants who are usually genetic counselling students in training who help us with doing intakes, which means contacting families before their appointments and gathering information. And we also work with other medical specialists in our multidisciplinary clinics. So generally, obviously, the doctors that work in the team are our clinical geneticists, and they usually will have training in paediatrics, general paediatrics or adult medicine, and then they go on to do specialist training in genetics. So we have a team of consultants who are the experienced geneticists, as well as a team of genetic fellows who are the geneticists in training. Generally, the doctors, if we are seeing a child, for example, who has a suspected genetic condition, generally they'll always see one of the doctors, one of the clinical geneticists or fellows for a medical consultation, because the doctors are obviously trained in making diagnoses. So they often will take a very detailed history from families about a child's growth and development. They will examine the child, looking for subtle features in that child that might suggest a particular genetic condition. They decide on what the appropriate genetic test is, if there is a test that's recommended. And if a diagnosis is made, they're, I guess, the people who can give information about what to expect in the future and recommend health surveillance as well. So obviously, if you make a diagnosis of a genetic condition and that condition might involve problems with the kidneys or heart, then the geneticist will make recommendations about follow-up or screening for that child. The genetic counsellors, I guess, are not so much involved in making a diagnosis, but we often see families, not always after, sometimes before, for consent, but generally after diagnosis, in terms of making, explaining things, providing that support, talking about genetic testing for other family members and so forth. But there definitely is some overlap in the roles, depending on what the condition is. So if we know that a child has a particular condition and they don't need a diagnosis made, then they would be more likely to see a genetic counsellor rather than a geneticist. I think I've talked about that. So that's sort of the slides that I've got talking about, I guess, the role of genetic counsellors. I don't know if anyone has any questions now, because the next part of my presentation is more about different types of genetic tests that are available. Oh, Joel? Yep.
Yeah, just purely out of interest, how come Victoria's got such a strong sort of genetic industry, for want of a better term, like they seem much more advanced in other states and how you guys are set up. I'm over in Adelaide.
Yeah.
We've got absolutely nothing like what you guys have over there. And even compared to New South Wales and Queensland, maybe you're fairly well set up, I'd say. Is there a reason for that, or just potluck, or someone was a good advocate back in the day? Or
I think it's probably historical, because the genetic services sort of started out of the Murdoch Children's Research Institute. Having said that, I think it's growing in other states like New South Wales, Queensland, South Australia, definitely growing. They've got their own laboratories. They've got fairly well-established services. Poor old Northern Territory, where we travel to, is I mean, they've obviously got a much smaller population, but they could definitely do with more support. But yeah, it's Victoria generally has I guess it's the history.
Yeah, it's definitely a bit of a hub, I think, at least in our eyes. Yeah, with people like Heather and some of the other NGOs and whatnot are sort of all based out of Victoria, which is good. Obviously, all the smart people are there.
There's a lot of smart people in Adelaide as well, I'll tell you.
It probably helps having the uni course based there. I'd say there's some correlation, isn't there? By having the uni based there, the people generally stay there or live there anyway.
Actually, with the UTS, sorry, I was going to say with UTS, that's now an online course. So the Melbourne Uni course is a face-to-face course. UTS have purposefully developed an online course because they were having the issue of, like, particularly states like Tasmania, they might have had someone who was interested in being a genetic counsellor, but, might have had children or couldn't travel to do a course for 2 years. And so they started UTS so that actually there are people in different states who will stay in those states as well, which I think is a good thing. Sorry, Heather.
Oh, no, I was just going to say with regards to the different states, I mean, I think having those big research institutes like the Garvan and MCRI really does make a difference. Elly, there was one question that Nadine asked, and it's more a comment that it was interesting to know about the saliva testing, as she was told that it isn't an option in Australia when tests are sent overseas. Are you able to talk to that point, please?
Oh, definitely some overseas labs now will accept saliva. So it depends a little bit on the laboratory, what test is being done. So if someone's having a whole genome sequence, which I'll talk about, that has to be done on blood. But if someone's having, for example, exome sequencing or a panel, often now it can be done on saliva if the laboratory that it's going to is accredited to extract and test DNA from saliva. But now, actually, we send a lot of, for the iGenetics clinic, we send a lot of samples to Finland, and they actually send the saliva kits to the patient's home address, and we send it straight back to Finland so people can collect it from home and just send it off.
Can I ask another question about that, just in brief? And you might touch on this and tell me so. In the case where it does go overseas, I'd like to know, how do you get the rights, or what's the consent further down the track if retesting needs to happen and that DNA sample is not in the country, and things around that area is also what I'm interested in. But yeah, with the saliva testing, it was to do with my daughter, and she's already had one blood test done about 5 years ago now. And we didn't have a follow-up since, and we didn't come up with anything. But yeah, that's why I was interested, because we know we need to do another one, and I don't think there is enough of her DNA to sort of do the next sort of stage of testing. And I wanted to know whether saliva or a sample would be suitable, and that's when it was referred to that they don't do it in Australia and they'd be sending it overseas, and they actually sort of suggested Sweden or Switzerland or something. I can't remember.
Yeah,
but yeah, so I was a little bit concerned. I would like to have it in Australia, and so that there will be continual sort of consent and access, and because the alignment of our health system would sort of allow for that. And I know we've got an Australian Genomic sort of alliance of some kind that will sort of protect us in that respect. But yeah, I wasn't really quite sure what happens with those other scenarios.
Yeah, it's a good question. And it depends a little bit on which lab overseas, it's going to. But you're right. I mean, having worked for Melbourne Genomics, one of their key objectives is to, you know, go back and reanalyze, store genomic data, go back and reanalyze. But because it's expensive, often clinical genetic services are making a decision about laboratories, obviously based on how good the laboratory is and how confident they are with that laboratory in terms of coming up with an answer, but also cost actually comes into it quite a bit because there's limited government funding for testing. And so they have to, I know at VCGS, we spend a lot of time weighing up cost of where to send samples as well.
But Elly,
Thank
it is
you.
difficult to track down your data when it's gone overseas. Having
Can
tried
be,
to
yeah.
track down
Yeah.
my own daughter's personal data,
Yeah.
DNA that went overseas, and there's no, you know, once it leaves Australian shores, it is hard to track. I mean, we're going back many years ago, but I think, you know, if you're consenting, you've got to be, you know, aware that who has ownership of that data once it leaves Australian shores.
Yeah, and also sometimes we have commercially sponsored tests. So sometimes companies like Novartis might sponsor a test, particularly in eye genetics as well, because there's now stem cell therapy for one of the eye genetic conditions. And so the drug company will sponsor genetic testing of the children because they want to find a child that they can sell their very expensive stem cell therapy to. But you know, you definitely have to be transparent when people are signing consents that their data could be shared for scientific research, or that it's, you know, going overseas, and exactly how that data will be shared and whether you can get that back.
Thank you. Thank you.
No worries. Joel?
Yeah, does Medicare pay for some genetic testing from memory?
Yes.
Yeah.
Yeah, it does. Yeah. So there's definitely Medicare item numbers for some genetic testing, particularly for there's what they call the childhood syndromes test, but the child has to be under 10 and must meet certain criteria.
Good to know, thanks.
Elly, Megan's got a comment. Do you recommend any private labs you recommend for families given wait lists at least 2 years at Monash?
Yeah.
And I know you can't recommend one over the other, but yeah, I mean, I think just because your child goes to Monash doesn't mean you need to access genetic services there.
Yeah, that's true. You can ask for a referral to a different genetic service. There's now private geneticists that are operating. And if you look on the HGSA website, the Human Genetic Society of Australasia website, it gives you a list of all the clinical genetic services in Australia. I mean, I think it's difficult. I think if you're looking at doing testing, it's better to do it through a geneticist or a genetics clinic, because it's not all the same in terms of the testing. And I can talk a bit more about this, but it's hard to know what to order without having a geneticist assessment. And so, yeah, I couldn't really specifically recommend a lab, but I can let you know that there are definitely options of genetic services. And you're right, if your child goes to Monash Hospital, doesn't necessarily mean you have to go to their genetic service.
Just put the link to HGSA's
Thanks.
for them in the chat.
So I'll just give a quick rundown of the different types of tests that are available. So most cells carry our DNA, which is our genome or our genetic material. And our chromosomes are these long strings of the DNA double helix that is found in the nucleus or the heart of the cell. And our genes are basically just sections along the DNA. And the sequencing techniques that we use basically read the building blocks of the DNA. So those DNA is made up of these 4-letter A, C, G, and T, and that's the code, the genetic code. And there's also a very small amount of DNA in our mitochondria. And our mitochondria are the parts of our cells that are like the energy generators of our cells. So I think of the genome kind of like a recipe book. The book is the whole genome. The chapters are the chromosomes, the sentences are the genes, and the letters are what we call the nucleotides, those A, T, C, and G. When someone has WES or whole exome sequencing or exome sequencing, that's only about 2% of the genome or the exome, and that's what we call the recipe that make the proteins for the body. So the exome are the genes that code for proteins in the body, or the known genes that code for proteins. And we obviously inherit our genes or our chromosomes from our parents. So generally our cells carry 2 copies of every single one of our chromosomes, and therefore we have 2 copies of every single one of our genes. The eggs and the sperm carry only 1 copy of each chromosome. And when the sperm meets the egg, the 2 sets of chromosomes come together into the new cell. And so therefore, a baby would have 2 copies of each chromosome, 1 from mum and 1 from dad. And changes that occur in the DNA from mum or dad could be inherited by the child. So that's kind of, mitochondrial inheritance is something that's a bit rarer. Basically, mitochondrial conditions, so conditions that occur in the DNA of the mitochondria, can only be passed down through the maternal line. They can't be passed down through dad's side of the family, but I won't go into that in too much detail because it's not that common. So when we're talking about genomic variants or genetic variants, they come in a range of sizes. So generally, a test that's not really done that often these days is a karyotype, and that was a test that was done basically looking to see whether there are any, whether the right number of chromosomes are present. There's the 2 copies of each of the chromosomes, and that there's no obvious changes in the chromosome number. The test that's done generally as the first line test now when a child has developmental delay, autism, is this test called a chromosome microarray. And that's a test, basically it's a test of dosage. So it's looking for what we call copy number variants, which means whether there's any extra or missing bits of genetic material in the chromosomes. And that test definitely can be done on saliva and is often ordered by paediatricians as a first-year test. The microarray can't pick up the condition Fragile X syndrome. So often you find that the chromosome microarray and Fragile X tests are done at the same time. So the other, so they're kind of large genetic changes. And then we look down to smaller changes which might affect single genes. So that might just be like a spelling mistake or a spelling error in the genetic instruction. Just like a G has swapped to an A, and those sort of genetic changes are generally picked up using sequencing technology. So using like a single gene test if there's a strong suspicion of a particular gene being involved, but that's not done that often these days. Often what's done is a panel test where they look at a group of genes at the same time, or an exome where they sequence the whole exome or the genes that we know that code for protein, or a genome where they're looking at everything, all of the genes, and even those that we don't know exactly what they're doing yet. And when I said before, there's different types of tests. So even if someone has an exome test, generally what happens there is that the lab sequences all of the genes, the 4,000 genes or so, but they might not analyze every single one of those genes. So an exome might have what's called targeted analysis, whereby the geneticist says, I want you to do an exome, but only analyze genes associated with intellectual disability or regression or epilepsy or so forth. So it depends a bit on what analysis is done on the exome as to which genes are looked at. This is just a picture of chromosomes under the microscope when they're stained. Generally, humans have 46 chromosomes, 23 pairs, one from each parent, and they're numbered from 1 to 22. And then generally males have an X and a Y, and females have 2 X chromosomes. And if you lay them all out like this is what a karyotype looked at before, looks like, which I mentioned before. And generally, chromosomes have 2 arms, a P arm, and I just think of that as P for petite, as like the short arm of the chromosome, and a Q arm, which is the long arm, and they're labelled in sections. Basically, you can see here there's P1.3, 1.2, blah, blah, blah. So sometimes when you see a microarray result, you might see something like there's a deletion of P11.2, which means that there's a bit missing on the short arm of this particular chromosome at that particular section. So it's like a map of chromosomes. And we know that different genes are located in different areas. So, for example, if we get a microarray result back that shows there's a deletion of this bit of this chromosome, we know to go in and look to see which genes are in that particular region and whether they are any important genes that we know if someone has a missing copy of that gene, that might have a particular consequence for that person's health. This slide here is an example of an aneuploidy, which basically means that there's 3 copies of chromosome 18 rather than the general 2 copies of chromosome 18. So that would be picked up on a karyotype. It would also be picked up on a microarray. That's a well-described genetic condition. So other types of chromosome changes might be things like a translocation, which means one part of the chromosome has swapped to another part. If it's a balanced translocation where there's no genetic material that's lost or gained, then that might not have any particular consequences for that person's health. But when that person has children themselves, if they pass on this chromosome here, that would mean that the child has an unbalanced amount of genetic material when they inherit the other copy of the chromosome from their parents. So you can have a balanced translocation, and then that can result in an unbalanced translocation in a child. A deletion is just like a bit missing of one of the chromosomes. A duplication just means that there's a doubling up of genetic material. And this here is an example of a chromosome microarray result, which, as I said before, is generally the first tier test that's done when there's a suspicion of a genetic condition. So this is a test, looks like this child has hearing impairment. And we can see here that there's a deletion on one copy of chromosome number 16 on the short arm of the chromosome at this particular point in the chromosome. So, as I said before, generally pediatricians can order microarray and Fragile X testing. It usually only takes a few weeks for the result and can be done on saliva, it doesn't have to be done on blood. Not that uncommonly, we get a result where we find a duplication or a deletion in a child of uncertain significance. So it might be something that hasn't been reported before. And often in that situation, we request parental samples to do a microarray on the parent, because if a parent has that same deletion or duplication and has no health issues themselves, often that means that it's just a benign or normal variation that's being passed on from parent to child that's not causing the health condition in the child. It's just a benign familial variant. So that's sort of microarray. And then when we go on, as I said before, and do further testing, there's the option of doing the single gene test, which is just like looking at one for genetic changes in one gene. A panel test where we look at several genes known to cause a particular condition. Exome sequencing, where we're testing all of the coding genes. And then genome sequencing, which will test all of the DNA, including the genes and the DNA in between genes, as well as the mitochondrial genome as well. I think it was Megan at the start explained that her child had had a test and the laboratory had found a variant of uncertain significance. So with when we do single gene testing, panel testing, exome or genome, we find lots and lots of changes in the DNA because that's what makes us different. And basically the lab used some complex bioinformatics to filter out which DNA changes are potentially causing a condition. They can discount many of them as benign or likely benign. Sometimes there is quite strong evidence that a particular genetic change is what we call pathogenic or a class 5 change, which means that it's causing that condition. And sometimes there's just not enough evidence in the scientific literature one way or the other. So we don't know whether a specific genetic change is benign or whether it's a disease causing or a pathogenic change. And that might be because it's never been reported before. That might be because there's some evidence pointing towards it being a disease causing change and some evidence pointing towards it being benign. And so it's sort of sitting on the fence in the middle and can be quite frustrating. Often it needs time to resolve. So maybe we usually review things again in a year or two to see if there's been any other cases in the literature where that particular change has been reported. Sometimes we test other members of the family to see whether or not they've got that same genetic change, because that might help us interpret it. Sometimes we just don't know and we have to wait and see. And so, yeah, if you have a likely pathogenic change, and that's a greater than 90% confidence that the variant is the cause, and likely benign is greater than 90% confidence that it's not the cause of the condition. And if it's benign, it's a greater than 99% confidence that that genetic change is causing the condition. Probably in the interest of time, I won't go into that. This is an example of an exome report that has been done. So I just thought I'd just talk through this a little bit. So this is a clinical exome analysis trio. So a trio means that 3 individuals have been tested. An individual and their 2 parents have been tested in 3. And often we do that because if the parents are unaffected by the condition, then that might help us filter genetic variants. So if they're present in the parents and they're new, and it's a new variant in the child, then that parent's DNA helps us act as a filter. I mentioned before about targeted analysis. So you can see here, it's an exome. So they sequenced all of the DNA, but the doctor requested targeted analysis. So they wanted genes that we know are associated with congenital heart defect, callosome, and these other intellectual disability and so forth. When you see the term Mendeliome, that actually means that they actually want all of the protein coding genes. So this is a very thorough analysis of the DNA. There's a result of this particular genetic condition, and it's telling us that this person who's tested has a pathogenic, which means a disease-causing variant in this particular gene, which causes this condition. And the trio analysis tells us that this genetic change is new in the child. It's de novo, which means it's happened, hasn't been passed on by either parent. I won't show it to you, but if you're interested in learning more, there's a video on the Learn Genomics site, which is part of Melbourne Genomics, which talks a bit more about genomic testing and the possible results and implications of testing and so forth. Heather just asked me to quickly touch on what the difference between research and clinical testing is. So clinical testing is done in a NATA accredited laboratory. You should receive the results in a time frame that should be quoted to you. So it might be 3 months, 6 months, 9 months, but generally you should expect to receive those results. And clinical testing shouldn't require an extra step of verification. So if you get a clinical test result from a NATA accredited laboratory, then that's a reliable test result. Research testing is something obviously that's pushing boundaries. It might be a test that's done in a research laboratory. It's not a clinical NATA accredited laboratory. It can take a long time. You might not expect to get a result for a year or 2 years, or, you know, there's generally not a time frame with research testing. And if someone is tested through a research study or a research laboratory and doesn't get a NATA accredited result, then the practice would be to confirm that result in a clinical or a NATA accredited laboratory. So that's kind of the main difference. And I guess the technology used for research testing might be newer technology that they're testing out. They might be looking for new genes, for example, for particular conditions. And I was just going to quickly mention the RD Now study that I know that Heather's been involved with. So we know that collectively rare diseases are actually quite common, can take a long time to get a diagnosis, you know, even when a diagnosis is made, few of these conditions have approved treatment options available. So there's a big research study called RD Now and another one now called UDN-Oz that's looking at discovery of new genes, as well as pushing the boundaries in terms of the type of testing that's available. So someone might have what we call a non-diagnostic exome. They might, as RD Now, go on to have a reanalysis, so another look at that data. If nothing's found there, they might go on and have a research trio exome. If nothing's found there, they might go on, if there's a really strong suspicion of a genetic condition, they might go on and have what we call a whole genome test and multi-omics approach. So one of the things now that is being done through the research study is something called RNA-seq, or also known as transcriptome analysis. And that's basically a technology that they're using to learn more about which genes are expressed or turned on in different types of cells and when and how these genes are expressed. So I won't go into the science of it too much, but basically RNA is copied from pieces of DNA and I guess contains the building blocks that go on to make the protein. So they're looking here at the order of these building blocks in making the protein. So that's something that you often hear people talking about only in a research setting at the moment. I think it's just on 9, so I might stop there. And sorry, I think I've gone on for way too long, Heather.
Oh, no, it's really interesting. That's great.